in a whole series of, this is Google's birthday present to Father Mendel when you're with his feet with Google. He did all these experiments, between 28 and 29,000 experiments on garden peas, gave 2 papers in 1865, published the paper in 1866, and it was just forgotten. There were 3 people that cited his work in the year between 1866 and 1899 or 1900, which would not have gotten you tenure anywhere. But he was a monk, and he had a steady job, and peas were just something else. Well, Mendel decided to ask simple questions. And this is the smart way to do science, to ask simple questions. And he did that. He did 29,000 experiments on common garden peas. He chose 7 simple traits. And these were either-or traits. From the seed shape and color, there's gray and smooth versus white and wrinkled. Seed interior color, green versus yellow. Flower color, purple or white. Right pod shape, inflated or peeled up. Constricted, unripe pod color, green and yellow. Flower location, either next to the stem or off on one of the tendrils. In front of the plant height, vine versus bush. If you are a gardener, bush beans, they will grow about that high, and then other beans grow up a trellis. Those are simple traits of either or. And this is kind of what it looked like, or looks like it in color. Get an idea as to what that stuff looks like. There are pictures in your book too, and a little different set of characteristics in your book, but let's just say there are 7. Now let's look at one of his experiments. What he did was for plant height, that is bush versus vine. He took a vine, and he took a bush, and he bred them, and he got 2 vines, 2 tall ones. And then he bred those 2, and he got, if you look at the ratio, about 3/4 of them were tall and 1/4 were short. Well, what happened was that the genetic influence that makes something short wasn't lost. You don't get medium-sized plants, you get either tall or short plants. You don't get intermediate. So that is not mixing, is it? Ronald Fisher decided to use a Latin square to show how this thing works. So let's take 2 parents up on top, white and red. Let's say the white parent has 2 genes for white, and the red parent has 2 genes for red. Well, if you breed them and look at the ratio, you get about 1/4 of the same as the next quarter, as the next quarter, as the next quarter. They're all the same, because the one parent is given one of the whites, and that's all that one parent is. The other parent is one of the reds, that's all that parent is. Let's say the red is tall and the white is bush. So, so we get these ratios, all of them are tall, because of red is we call dominant. Describe that. The white is recessive, when tall is dominant and short is recessive. Now, let's breed the second generation, the ones that have the 2 different alleles. We can do that with plants because incest is okay in plants. It's okay. Well, what happens is you get let's say male hair or female hair, or male hair and female hair, doesn't matter which. And half of the time, each one of those parents will have a red allele that will pass on. And so red and red, you get red. Red and white, you get red. white and red, you get red. White and white, you get white. About a quarter of the time, you get that bush, or recessive, we call it, as opposed to the red, which is dominant. We're going to go back to this picture time and time again. Let's first of all look at the cell. There are a couple of things to point out in the cell. Obviously, the nucleus is the important one, the blob over here, there. But there's also the mitochondrion right up here. And that, we're going to talk about a little bit later, that contains genes which are passed from mother to child. They pass to both sons and daughters, but only daughters can pass it on because of the size of the ovum as opposed to the size of the sperm. Over here we have ribosomes. We're going to talk about those a little bit later because they take an important role in producing what we are from the genes. So let's look at the nucleus next. And that's, the nucleus has a lot of so-called chromatin. And what a lot of that chromatin is are chromosomes. This is a chromosome, a Photoshop picture of a chromosome. It has two sides, two chromatids. This is what the chromosome looks like. And schematic, it has two chromatids. It has a centromere in the middle, which binds it together. So this is the simpler picture. Let's look at a little more complex picture. This is a chromosome. It's got the centromere, and it has two arms, a short arm on top and a long arm on the bottom. And the short arm on top is called the P arm, and the long arm on the bottom is called the Q arm. And we use those to describe where specific genes are on chromosomes. So we'll say it is on 7p3.1. That means it's the seventh chromosome. P is the short arm, and it's zone 3 and position 1, or zone 1. Notice that there are other things at the ends of the chromosomes called telomeres. These help in the process of both meiosis and mitosis. And we have found out also that they are extremely important in how long you live. If your telomeres are in pretty good shape for years and years and years, you're going to live a long time. If your telomeres start to break up and disintegrate, you will probably die in your 40s, 50s, or 60s instead of in your 80s or 90s. The telomeres are very interesting. I'm not quite sure why that is. It's probably because if the telomeres are not in good shape, meiosis is not going to keep the body renewing itself very efficiently. And this is a picture of a chromosome, electron microscope picture of a chromosome that hasn't been photoshopped. But it looks just like what happens when your cat gets a hold of a ball of yarn. And what it is, it's just a lot of DNA. We're going to talk about very briefly. Now, if we look at the chromosome is a word that just means colored body. Chromo, color, some somatic body, colored body. And these stain differently. So these are the human chromosomes. We already looked at the human and the chimpanzee chromosomes to show that they're very similar. And the human number 2 chromosome is made up of 2 of the chimpanzee chromosomes, 2A and 2B. This is, unfortunately, not a clear picture of it. The dotted line runs through the centromere, and you can see there's the P portion and the Q portion on each of the chromosomes. We have 22 pairs of chromosomes that are the same in terms of where genes are. They match up perfectly. Females have another pair here that match up perfectly, and they are the X and the X. Males have 2 chromosomes like the X and X. One is an X, but one is a Y. and it's the Y chromosome that makes males male.